Good morning, everyone. I am delighted to welcome you all today. Today, we will be exploring an important topic, rotavirus. My name is Dr. Mayuri Rani, and I will guide you through this presentation. Over the next few minutes, we'll cover what rotavirus is, its impact, and how we can prevent it. So let's get started. Rotavirus, when viewed under an electron microscope, has a very distinctive wheel-like appearance. This unique shape is actually how it gets its name. The word rotavirus comes from the Latin word rota, which means wheel, highlighting this characteristic look. Now, in terms of structure, rotaviruses are non-enveloped, which means they lack a lipid membrane surrounding them. Instead, they have a triple-layered capsid, or protein coat, that surrounds their genetic material. This layered structure is important because it provides stability and protection to the viral RNA inside. Speaking of genetic material, rotaviruses have a segmented RNA genome, meaning their genetic information is divided into multiple pieces rather than one continuous strand. This segmentation plays a role in their ability to evolve and sometimes reassort, which can impact how we approach treatment and prevention. Overall, these features, its wheel-like shape, layered capsid, and segmented RNA, are key to understanding how rotavirus infects cells and why it's such a resilient pathogen. Let's take a closer look at this transmission electron micrograph of intact rotavirus particles. As you can see, this digitally colorized image helps us visualize the actual structure of these viral particles. The detailed imagery allows us to observe the virions, which are essentially the infectious units of the virus. Understanding their structure is crucial because it gives us insights into how the virus infects cells and how we might target it with vaccines or treatments. This image connects to our overall theme by illustrating the complex architecture that underpins rotavirus's ability to cause disease. Recognizing these structural features is fundamental in developing effective strategies to combat infections caused by this virus. Rotaviruses are quite small, about 70 nanometers in size, and they're non-enveloped RNA viruses, which means they lack a lipid envelope surrounding their structure. They belong to the family Reoviridae, a group that includes several other double-stranded RNA viruses. The core of the virus, called the nucleocapsid, is pretty complex. It's made up of 3 concentric shells that tightly enclose the viral genetic material, specifically 11 segments of double-stranded RNA. This layered structure is essential for protecting the viral RNA and playing a role in the virus's replication process. Now focusing on the outermost layer, the one that interacts with host cells, this contains 2 key structural proteins known as viral proteins or VPs. The first is VP4, which is a protease-cleaved protein, often called the P protein. The second is VP7, a glycoprotein, also known as the G protein. These 2 proteins are particularly important because they define the serotype of the virus, meaning they determine the specific immune response the virus will provoke. That's why they're so critical in vaccine development. They're the primary targets for neutralizing antibodies, immune proteins capable of blocking infection. Essentially, by recognizing these proteins, our immune system can prevent the virus from causing disease. So, understanding the antigenic structure of these proteins helps us grasp how vaccines work and why they're effective. It's a great example of how detailed structural knowledge directly informs our strategies for protection against rotavirus infections. Because the 2 gene segments that encode these proteins can segregate independently, a typing system consisting of both P and G types has been developed. This means that each segment can vary separately, so to accurately identify and classify these viruses, we need a system that considers both gene types. Although characterizing G serotypes using traditional methods is fairly straightforward, it's a different story for P serotypes. These are more challenging to determine with conventional techniques, which is why we now rely heavily on molecular methods. Specifically, we use nucleotide sequencing to define genetically distinct P genotypes. These genotypes usually match well with the known serotypes, but to distinguish them clearly, we put them in brackets, like P8. This helps us differentiate genetic types from those identified simply through antigenic analysis, which is more traditional. Overall, this typing system allows us to better understand the genetic diversity and epidemiology of these viruses, which is crucial for accurate diagnosis and vaccine development. Rotaviruses are classified into 7 different serogroups, labeled A through G. This classification is based on the antigenic specificity of their capsid proteins, which are key to how the immune system recognizes the virus. Additionally, the classification considers the pattern of electrophoretic mobility of the 11 RNA segments in the viral genome, which provides a genetic basis for distinguishing these groups. Of these 7 serogroups, only groups A, B, and C are known to infect humans. Among them, Group A rotaviruses are particularly significant because they are responsible for causing severe, life-threatening disease in children worldwide. This highlights the importance of understanding and targeting group of viruses in our efforts to prevent and control rotavirus infections. So, in summary, the classification helps us understand the diversity of rotaviruses, their potential to infect different hosts, and especially emphasizes the global health impact of Group A rotaviruses in children. This knowledge is crucial for developing effective vaccines and public health strategies. Rotavirus disease typically presents with vomiting and watery diarrhea that can last from 3 to 8 days. These are the hallmark symptoms that help us identify the infection. In addition, many children also experience fever and abdominal pain, which can make them feel quite uncomfortable. You might also notice a loss of appetite and signs of dehydration, which are very important to recognize early. Dehydration symptoms include decreased urination, dry mouth and throat, feeling dizzy when standing up, crying with few or no tears, and sometimes unusual sleepiness or fussiness. Understanding these clinical features is crucial because they help us assess the severity of the disease and determine the appropriate care. Recognizing dehydration early allows us to intervene promptly and prevent serious complications. This ties into the overall theme of managing rotavirus effectively and highlights why early detection is so vital. The incubation period for rotavirus disease is approximately 2 days, which means symptoms can start showing quite soon after a child is exposed. It's important to note that children can actually get rotavirus more than once because neither the vaccine nor natural infection provides complete immunity. So, even if a child has had it before, they're still at risk of future infections. In fact, the first infection tends to be the most severe, causing the most intense symptoms. This is why prevention and early treatment are so crucial. Remember, understanding these points helps us better grasp why rotavirus remains a significant concern, especially in young children who are most vulnerable. The primary way rotavirus is transmitted is through the fecal-oral route, which usually happens when people come into direct contact with one another. Because the virus is quite stable in the environment, it can also spread through ingestion of contaminated water or food, and by touching surfaces or objects that are contaminated. In temperate regions like the United States, we see a seasonal pattern, with most cases occurring during the winter and spring months, and outbreaks typically happen from January through June. It's important to note that the highest rates of illness are among infants and children under the age of 5. While adults can also contract rotavirus, their cases tend to be milder. This epidemiology pattern helps us understand the importance of prevention efforts, especially in young children, to control the spread of the virus and protect vulnerable populations. Now, let's talk about the different methods used for diagnosing rotavirus infection. One of the most sensitive techniques is nucleic acid detection (PCR) assays, which can be used specifically for rotavirus alone or as part of multipathogen panels. This approach allows us to detect the virus's genetic material with high accuracy, and it's especially useful in surveillance testing or research settings. Another rapid diagnostic method is antigen detection immunoassays performed on stool specimens. These tests are valuable because they give quick results, helping clinicians make timely decisions. In addition, Characterization of rotavirus strains is crucial for understanding the circulating types and tracking outbreaks. This is typically done through nucleic acid sequencing, performed by laboratories engaged in surveillance or research. Overall, these diagnostic tools, PCR assays, antigen tests, and strain characterization, are essential components in our effort to accurately diagnose rotavirus infections and monitor their spread. When it comes to people with healthy immune systems, rotavirus disease is usually self-limited, meaning it only lasts a few days. It tends to resolve on its own without the need for specific medications. The main focus of treatment is supportive care, particularly oral rehydration therapy. This involves giving fluids orally to prevent dehydration, which is the most common and serious complication of the illness. By maintaining proper hydration, we can help the body recover more comfortably and avoid additional health issues. This approach highlights the importance of simple, effective interventions in managing rotavirus infections, especially in otherwise healthy individuals. The CDC recommends that all infants receive routine vaccination against rotavirus to protect them from severe diarrhea and dehydration. There are 2 vaccines available for this purpose: Rota, also known as RV5, which is a pentavalent vaccine that includes 5 rotavirus strains. This vaccine was licensed back in 2006 and is administered in 3 doses at 2, 4, and 6 months of age. The other option is Rotarix, or RV1, which is a monovalent vaccine targeting a single rotavirus strain. It was licensed in 2008 and is given in 2 doses at 2 and 4 months. The key takeaway here is that vaccinating infants early and on schedule is crucial for preventing rotavirus infections, which can lead to severe illness. These vaccines are part of the overall effort to protect children and reduce hospitalizations related to rotavirus. By following the recommended immunization schedule, we help ensure that infants are best protected during their most vulnerable months.